Hi everyone. Thank you for joining us again this week for our weekly sort of Exploration Club webinar. So in today's sort of webinar, I'm really excited to sort of show you the potential and really present on a research or a promising sort of pair of blood-based markers and how this could aid the diagnosis of Alzheimer's disease. So that will be our focus on today, sort of neurological diseases. And just for anyone in the team or any scientists who are joining us for this webinar and they're new to bioterrain or they're not working on sort of central nervous system diseases. So Alzheimer's, just as a recap, it is a sort of devastating neurodegenerative disease that affects millions worldwide. And early detection is really crucial when it comes to effective management in these patients, and also mainly because current diagnostic methods can be invasive and expensive. So this will sort of be the focus. And to give you an idea of how we'll be able to really answer very specific questions for this case study, we'll be mainly sort of navigating the biotrain ecosystem, but mainly focusing on the talk to data repository. So both for single cell RNA-seq and spatial transcriptomics data. Okay. So a bit of background, again, if any scientists who are joining, they're not really working on this area, but would be a potential area. So Alzheimer's disease is sort of the most common form of dementia, and it is really characterized by progressive decline in cognitive function. And as depicted in this sort of figure that I'm showing now, sort of Alzheimer's disease is associated with several pathological hallmarks in the brain. And these hallmarks are really defined into sort of five main categories. So we can see, for example, that in the sort of brain atrophy side, which is the Alzheimer's disease, as compared to what would be normal sort of development and sort of central nervous system processes, the amyloid beta plaques, these are some sort of hallmarks of Alzheimer's disease, which are showing sort of these abnormal protein deposits that are accumulated between the neurons. We can also see neurofibrillary tangles. So these are twisted filaments of protein called tau, which are found inside of neurons. And then there's also synapse loss, which is sort of defined as Alzheimer's disease, which leads to significant loss of connections between neurons and impairing communication. So those are sort of some of the main ones. And we can also see that some other hallmarks in Alzheimer's disease is brain atrophy, which is mainly defined by the overall size of the brain and how it shrinks due to neuronal death. And also dystrophic neuritis, which are damaged nerve fibers that can contribute to cognitive decline. So these are sort of main hallmarks that are common and that really characterize sort of this disease. And additionally, the sort of slide highlights the role of glial cells as well. So glial cells are really important, including astrocytes and microglial cells in Alzheimer's disease. And why that is the case, so these cells play a crucial role in the immune response and in inflammation. And these are associated with this disease, which will be very important as we go through the session today and how we'll be analyzing these cell types with interactions with other blood-based markers. Okay. So while the brain sort of tissue analysis remains sort of the gold, I guess, standard for diagnosing Alzheimer's disease, this could be very sort of invasive, and these methods really are limited by their widespread use. Fortunately, there is a lot of extensive research that is being sort of conducted, which is trying to identify very specific, promising blood-based biomarkers that offer a less invasive and more sort of accessible approach to diagnosis. And studies have shown that PBMCs particularly contain biomarkers associated with Alzheimer's disease, such as this amyloid sort of beta, tau, and also other inflammatory markers as well. So these could be sort of potentially used to predict the development of Alzheimer's disease or sort of the development of how to monitor disease progression. And really, by exploring these potential blood-based biomarkers, which is sort of the focus of the session today, we'll try to aim and develop how accessible and convenient these diagnostic tools are in Alzheimer's disease. Okay. So now that we sort of discussed a bit of background on Alzheimer's disease, sort of the hallmarks in the area, and also potential use of PBMCs as a source of biomarkers, we'll try to really delve specifically into this question: how can we explore potential marker genes for Alzheimer's disease using PBMC samples? So really harnessing all of the public data that we have in the Talk to Data system and being able to identify these marker genes from different PBMC samples. Okay, and since this will be done on the Talk to Data system, I'll also show you how to navigate the system and identify these studies. So when you go into the Biotrain ecosystem, and if anyone in the sort of webinar is new, they haven't used Biotrain before, but they got this link to the webinar, you could reach out to support, and any colleagues in the team will be able to share that link. So you're able to really try the system. So when you log in into Biotrain, you'll directly go into the Talk to Data system, and you'll see that this is broken down by single cell RNA-seq and different spatial transcriptomics technologies databases. So this is where we harness and where we our repository of public data. So you'll need to go into the Studies tab, and within the Studies tab, this is where you can search either for the title of a paper that you might want to use as an example, or it might be that you're focusing on only PBMCs across different diseases. Then you can use the filtering standardized ontology to really define what other studies have that criteria. But for this case, we'll be looking at a very particular study, and I'll explain why we're concentrating on this study specifically. So when you search for the study ID, which can be found on other repositories, you'll be able to then identify the data set and then be able to explore that study further. So this will be the study that we'll be focusing on and really trying to answer that identification of marker genes and blood-based markers. Okay. So I'll go back to the slides. So a bit more as to why we're really focusing on this study. So in this study, the authors utilized a data set of PBMC samples from individuals with or without Alzheimer's disease, and they applied single-cell RNA-seq to these PBMCs that were isolated. And this sort of was in the aim to really identify distinct immune cell populations and associated gene expression patterns in Alzheimer's disease. So our goal now, in terms of harnessing the public data that is available, we'll try to identify specific biomarkers that are differentially expressed between these individuals with or without the disease. And by analyzing these differences, we really hope to find very new insights into the underlying biological processes involved in AD. Okay. So I'll go into the next slide. So in using single cell RNA-seq sort of data, and I'll sort of explain a bit more on this slide. So we conducted a differential gene expression analysis to really identify these genes that are differentially or significantly upregulated or downregulated in these PBMCs from both AD patients and the healthy control donors. So you can see that in the volcano plot, which is just a way that we visualize the different genes and how they're sort of differentially expressed. So if we explain a bit more, so what we were able to identify is that within the volcano plot, there are two particular marker genes that are significantly sort of emerged and are differentially expressed within the groups. And these two genes are FCER1G and TYROBP. So these particular markers are both significantly upregulated, as you can see, in Alzheimer's disease only, so not really the healthy control samples. And a way to really be able to then run this analysis, so if you're just starting to use it and you want to sort of create these plots from the data, when you explore the data set directly on the system, you'll be able to then go into the differential gene expression feature. And this will allow you to either run a two-group comparison. So in this case, I'm not particularly focusing on specific groups or cell types. I'm only interested in all of the genes or marker genes within healthy donor samples and Alzheimer's disease. So when you choose a two-group comparison, you'll have different ways on really how to lasso the cells. And this really depends, again, on the question that you're answering. But in the metadata, you can actually select the actual disease samples. So in any study or public study, you'll be able to find that metadata, and you would be able to then select what samples you're trying to analyze. So in the Alzheimer's disease, this would be sort of our first group. Our second group would be all of the remaining cells. But again, you have to make sure that it is selecting the correct cell group. So I'll select then the normal control samples. So these would be control. And once you have these two groups selected, there's a lot of other steps that you can add. And again, this really depends on the question that you're trying to answer and what statistical method you're going to apply. So once you have those parameters defined, you can then run the analysis, and this will create sort of that specific map that I showed you, the volcano plot, and also other downstream analysis that you would be able to perform from there. Okay. So I'll go back into sort of the slide. So this is what I wanted to show you, how you can actually run it very quickly, and it's able to calculate those results as well. Okay. So if we go into sort of the next analysis, which is looking at the heat map and specific expression of these two genes across different cell types. So you can see here we generated a heat map. And how are you able to do that on the study? So when you generate the differential expression analysis, you can then go into actually generating the heat map where you can add other clusters from other metadata. So this is where you would find the other downstream features. And this is where our team sort of analyzed this data. So what is this data showing us? So by gaining a better understanding on how the cellular context of FCER1G and TYROBP expression, we examined really the distribution across different immune cell types. And as you can see here shown in the heat map, both these two marker genes are highly expressed mainly in natural killer cells, particularly in Alzheimer's disease patients. So this finding suggests that there are alterations in NK cell function that may be involved in the sort of pathogenesis of Alzheimer's disease. And into sort of further investigating the role of NK cells, you can also identify other associated genes that could provide more valuable insight into sort of developing targeted tools, or if you need to develop immune modulated treatments, you would need to be able to get a bit more information on other marker genes as well. So this is just an initial part of the analysis. So you're able to really visualize that clearly across the groups and also based on the cell types, which is the clustering effect here at the very top. Okay. So I'll go into sort of the next step. So literature really, if we sort of apply this analysis that we found in these results from this data set, so other literature suggests that FCER1G and TYROBP play an intriguing role in the context of Alzheimer's disease. And how is this sort of role sort of defined? So FCER1G was found to be enriched mainly in the necrosis pathway within the p-tau interactome. And this suggests a potential role in neuronal death and inflammation, which are key features of Alzheimer's disease. And again, this was at the initial start of the webinar, which I showed you all of the different hallmarks based on both atrophy and other molecular processes in the brain. So we can really see those correlations from other literature as well. And the gene may contribute to both degenerative processes associated with the disease. So we really need to be able to find sort of the functional effects as well and what pathways. So besides FCER1G and TYROBP, we found that these two sort of markers act as an adapter protein involved in the activation of innate lymphoid cells. And this finding suggests that the role of immune responses that contribute to sort of Alzheimer's disease pathology, such as inflammation and clearance of amyloid B plaques, can be sort of involved from these roles. So these are all sort of past literature that are really sort of, I guess, mirroring what we're able to define and find on our study in the analysis. Okay. So I guess the next step from here is how about FCER1G and TYROBP, and how are they distributed in the brain of the Alzheimer's disease patients? So this is really not only using single cell RNA-seq, but it's really trying to understand sort of the spatial distribution of these genes and if there's any other spatial variable genes that we're able to identify as well within sort of the brain and different structures or different layers of the brain as well in these patients. So you are able to really answer this question as well from talk to data. And as I mentioned, when you navigate the talk to data system, you are able to choose exactly what technology you want to sort of find what studies in the repository we have. So we can go to bulk spatial transcriptomics or single cell spatial, which has a better resolution. But it is limited by the number of markers or genes that are used in the panel. So you really need to sort of define what your question is, and if you're just trying to see the spatial distribution of the gene as well. So in the bulk transcriptomics data set, you can go into studies, and this will give you an idea of how to filter as well by different criteria. So since we're only focusing on Alzheimer's disease, I can go to sort of the label that is showing us studies within that area, and we can then search for those studies. So you can see here that we have this particular sort of bulk spatial data set that is looking at sort of genes associated with vulnerable sort of location in the middle temporal gyrus of these AD patients. So the next step is to really explore the study further on Lens. And this is our Lens platform, which is mainly for spatial transcriptomics analysis, where you can query a specific gene and you can see sort of the distribution of the gene further. So the first step, I'll go back into sort of the slides to really explain what we're going to do in the analysis. So in order to really sort of investigate the distribution of these two particular genes using spatial transcriptomics, so unfortunately, due to the invasive nature of the brain tissue analysis, direct measurements of these gene expression in Alzheimer's disease brains are really limited. So there's not several studies that are really trying to understand spatial distribution or spatial variable genes in the brain tissue. And however, there is some existing literature that does suggest that these genes may have a sort of differentially expressed role in specific brain regions associated with Alzheimer's disease, and particularly more so in the hippocampus and in the cerebral cortex. So to really understand the spatial distribution of these marker genes in the brain, we'll utilize that spatial transcriptomics data set to really understand how it relates to the middle temporal gyrus of these AD patients. And this data set was generated, just so you know, using the Visium platform. But again, there's more technologies that we carry, Visium HD, Cosmix, 10xinium. So there's a lot of other data sets or technologies that you can use to really answer this question and really try to understand early stage Alzheimer's disease and late stage Alzheimer's disease samples. So if I go back to sort of the data set that we have here, we can then query any particular genes. And again, these are our two genes of interest that we've been focusing on from the single cell RNA-seq data. So when we search for the expression of these two genes, and I zoom in so you're able to really see that, you can see both the spots. So when it comes to Visium data, it is limited by how the resolution is sort of located in the sample. So you're not really able to see like the cell nucleus or the single cell resolution on the image, but what you're seeing is the spots that are sequenced in the tissue. So you're able to see what spots are particularly expressing each sort of marker gene and which spots are co-expressing both at the same time, which is quite important if you're interested in co-target effects and looking at co-expression. So from here, if we want to further understand this, like how does this relate to the actual samples, you can always overlay other sort of information from the study. So it could be specific layers in the brain or the actual conditions. So if I zoom out again, you can clearly see which are the controls and which are the Alzheimer's disease. So when I search for them, what we're able to see is that the expression of these two targets are higher in these AD patients. So these are the AD samples. So we are able to see a higher sort of target effect and also the expression of these two genes higher. And however, to make sure that this is statistically significant, we can also conduct differential expression analysis between the two groups to really identify if these two markers are significantly sort of upregulated only in the Alzheimer's disease samples. So in order to do that, again, it has a similar step where you would have to go to that feature. But since we covered that in the single cell RNA-seq, I'll go directly to the analysis that we were able to find. Okay, so as shown from this spatial transcriptomics data set, this sort of study showed within the volcano plot that these two markers are significantly enriched in the AD brain specifically. So specific spots that are sequenced across the brain in these patients as compared to control. So again, we're able to really replicate what we saw from the single cell RNA-seq data and really able to see it, how it's spatially distributed in the brain. So it's showing sort of consistent results. And other findings that we're able to also see is that this really suggests that these marker genes that are sort of expressed are common features of Alzheimer's disease that are occurring both at the peripheral and also the central nervous system. So really specific regions of the brain that sort of these genes can be targeted. Okay. So what else can we gather from this? So based on the differential expression analysis results, we can also perform further enrichment pathway analysis to really uncover what pathways are involved or enriched using these two targets. So in order to do that, if I go back into the study in Bioterrain Lens, when you perform the differential expression analysis, and again, you can go through the result that you have at any point, you would then go to the pathway analysis to be able to see those pathways that are enriched. This might take a few seconds just because it's trying to calculate it based on all pathways and also the specific marker genes within each of the pathways across the two groups. So in order to show you here, I had sort of the run of this analysis. And what is this sort of result showing us? So in the pathway enrichment analysis, this revealed that both the FCER1G and the TYROP are significantly enriched pathways related to the gliosis, which is a process involved in activation and also function of microglial cells. So if we take a step back, when we started the initial webinar, I mentioned another hallmark of Alzheimer's disease was looking at microglial cells. So what this is showing us is that, again, we can also validate that these two markers are involved, not only in the peripheral sense, but also in the spatial distribution of different layers and how they're upregulated within different regions of the brain. And we're able to see that this also has an effect in very specific sort of pathways, as we see here with the enrichment result and on sort of the particular genes that we're able to sort of analyze further. So these very important blood-based markers. Okay, so microglial cells, if you're not really or you haven't really worked with these cells before, so these cells are quite crucial when it comes to immune response within the brain. And their dysfunction has been implicated in various neurodegenerative diseases, as we can see with the case of Alzheimer's disease. So the enrichment of these two genes in this particular pathway suggests that these genes may play a role in regulating microglial activation and function when it comes to Alzheimer's disease pathology. Okay. So this spatial data set really focused on the middle temporal gyrus, which is a vulnerable brain region in Alzheimer's disease. And to really gain a precise understanding of the spatial distribution of these particular two markers, and also looking into within sort of these different MTG regions, so different layers with molecular sort of layer information, we can then examine very specific cortical layers and subregions that express those genes as well. So how can we perform that? So if we go into a bit more on how we're able to really understand this. So if we identify the specific cortical layers within these different sort of molecular regions in the brain or in the samples, the authors within the study employed a combination of methods to really define the layers and to annotate them on the spatial sort of distribution slides. So they used sort of three main methods. So clustering analysis, which was a technique used to group the cells based on their gene expression profiles, which allowed to really identify the distinct cell populations corresponding to different cortical regions. They also used immunofluorescence staining as well, which allowed to define the specific cell type markers, which were used to conform and identify the different cortical layers as well. So by combining these two approaches, the authors were able to reliably annotate the cortical layers in the spatial sort of data set. And this gave us sort of this accurate annotation, which is essential when we're trying to map them based on these blood-based markers. So if these have any particular role based on sort of the regions within these MTG sort of defined layers. Okay, so in order to do that on the study, very quick, I'll show you if I go back to the data set here. In order to do that, what you can do is perform a composition analysis, which is just a way to map either the composition or the proportional changes of the marker genes and how they're expressed. So in composition analysis, again, you'll have to select what sort of information you're trying to map it to. So since we are working from the layers that the authors defined, we can then map how these two genes are expressed across different layers of the brain within the sort of lens system. And we did perform this and sort of zoomed in so you're able to see it a bit more clearly. So what can we see from this analysis? So in the composition analysis, this reveals that FCER1G and the TYROBP sort of marker gene is highly expressed in specific cortical layers within the MTG. These both genes are found to be enriched in layer 1 and in white matter. So you can see here, this is based on the color scale of that expression of TPM and in the white matter as well. So it's not really based on just I guess the size of the nod. The nod is really the percentage of cells within that region that were defined with that sort of annotation. So it also has a high expression, even though it's quite small to see. It just means there's less cells within the white matter. Okay. So what else could we gather from this as well? So the white matter is composed of primarily myelinated axons, which are connections to the neurons across different brain regions. And during these layer identification process, the author also used specifically in layer 1 the annotation of SPRAC and CXCL14. So for SPARC, which is another important marker, this is sort of used in matrix-associated protein, which is involved in the extracellular matrix synthesis and cell sort of shape changes. And in terms of CXCL14, this is sort of a cytokine gene, which is involved in immune cell migration and also activated NK cells. So again, this connects us to sort of the single cell RNA-seq data in terms of how these blood-based markers were particularly upregulated in NK cells. And we're able to really see that cross relationship when it comes to other markers that were also labeled by the author for these layer 1s and also important markers in the white matter. So you're really able to see sort of this co-regulation effects as well. Okay, so in terms of what we were able to learn from this potential case study and looking into blood-based markers, not only from single-cell RNA-seq data, but how that applies into spatial distributed sort of imaging data. So there's three particular things that we learned from today. So FCER1G and TYROBP are two potential markers or marker genes for distinguishing AD from normal healthy donor samples. So we were able to really map this across different types of data and different types of sort of functionalities that we were able to use. These two particular markers were found to be expressed in the immune cells with the highest expression in NK cells in PBMC samples. And when we tried to really correlate that in the spatial transcriptomics data set, we were able to see that the authors also were able to find other co-regulated genes that were used to annotate layer 1, which is consistent with also sort of these two blood-based markers and in the white matter brain region as well. So these two marker genes were also found to be involved in the microgliosis, and this was confirmed when we ran the pathway enrichment in the spatial sort of distribution or transcriptomics data set, where we were able to map both the location of these blood-based markers in specific regions, but also involved in this particular pathway. So this is quite important, and it's really showing us this enrichment in Alzheimer's disease. So it's really hallmarks within the disease pathology. Okay, so this is sort of concludes what we were able to learn and how we can apply that using the talk to data system. And I believe my colleague has sent over sort of a poll to get some feedback. And I highly recommend you send us as much feedback as you can, because it really drives what case studies we're doing in the next weekly sessions. And it really helps us know if this is really helpful, or if you would want it sort of in a different way, or you want us to explain sort of features in other types of case studies. So feedback is very important. So I'll go into the Q&A session now just to be able to answer any questions that others in the team have as well. So one of the questions I see here is, do you see the same expression pattern in other neurodegenerative diseases? So in order to really know that, we would need to investigate it further, maybe in creating a custom atlas comparing other neurodegenerative diseases and being able to map if these blood-based markers are, again, in effect, upregulating within these different neurodegenerative diseases. So in order to really do that, what you can do, if I show you very quick, so if we go into the talk to data system again and we explore it, one way that you can create an atlas where you're including other neurodegenerative diseases. So in studies, you can go into the filtering tab, and this is where you can go into condition, which is just a standardized label that allows you to really select particular sort of data sets within that. So again, it really depends like what cell type you're using or what condition you're going to focus on. But we include other studies, not only in Alzheimer's disease, and especially when it comes to single cell RNA-seq data, you can go through the list and, for example, select that you want to look at like demyelinating disease, other neurodegenerative diseases, Parkinson's, and you can then include these as one custom atlas where you're going to compare these blood-based markers and if these could be potential sort of markers for these other conditions as well. So in order to create it and create the integrated atlas, you would have to go to create a custom data set. And this is where you'll see all of these selected cells across these different studies that were matching the condition label that was filtered. Okay. So that I hope answers one of the questions from the users. The other question is how expression of FCER1G and TYROP in NK cells is related to microgliosis. So if we go back into some of the results that we saw, so these two genes were always expressed, or are these genes always expressed together? So in order to really do that, what you can do is try to search in Talk to Data using the gene search, and try to see if, in effect, these have any sort of co-expression matrix, and if you're able to really confirm if these blood-based markers are always co-expressed across different diseases. So in order to do that, if you go into Talk to Data, you would just go into the genes category, and this is where you can input the two genes to really see if there's any sort of co-regulation effect that are sort of expressing. So I'll add the FCER1G and then the TYROBP. Okay. And once you have those genes, you can search and it'll generate that ontology map on how these genes are expressed across what cell types and if they're co-expressing as well, and at what level are they co-expressing. So this is where you would be able to really answer that question as well if you're interested in sort of co-regulation. So it also looks like the database of spatial data is quite limited. Is there any plan to expand it? So yes, there is a plan to expand it. So we are running I guess from other webinars that we've had already, we are running a public curation, a high public curation program for Visium HD data and also other types of spatial data if you're interested in integrating that type of data, and also RNA bulk. So if you do have studies that you're interested in really using to integrate it into an atlas or to just use it to sort of get more information on your marker genes that you're targeting, you can send us what studies you want us to curate across either spatial, single cell RNA-seq, or bulk RNA-seq. And a way to do that is whenever you're looking for studies and Talk to Data, you'll also have this tab where you can request a study to us. So when you go into the request management part, you can then request, and we'll know from what account it's coming from, or who's the user that is accessing the account and what studies they want us to curate. So we'll be able to update you once that curation is finalized and how you can visualize the data further. You can also view early access data sets. So these are all data sets that are not yet released in the Talk to Data system, but these are studies that other users might have requested before, and you can also add them to your request. So it just saves you time instead of waiting for us to let you know it's already available. So you can look at and search if there's any studies that would be within your area as well. Okay. So I think those are most of the questions that I see. So is Talk to Data available for free? So we do offer, Mohammed, a sort of, I guess, a trial license if you want us to sort of, if you want to get an idea on how powerful the system is. So you can reach out to us and we can give you access as a trial period to Talk to Data or to any of the other modules if you're working on your own data as well and you want to upload it into the system. We do have platforms for that as well. So these platforms like bBrowser X, Lens, or BioStudio would be mainly if you're uploading internal data set into the system. So just reach out. We'll give you an idea on the trial period. And if you want to then continue using it for your data or for a public data access, we can then discuss that further as well. Okay. And I think those were most of the questions from the Q&A. If there is other questions that you want to ask us more like privately because it's relating to your data, you can reach out at support@bioterrain.com, and that will connect us either to the development team if it's a feature you want us to develop in the system, or if you just want to be able to collaborate and get an idea on how to use the system for your analysis. So yes, just reach out to us at any time. We're always happy to support. Okay, so thank you everyone for joining us today. I'll give a few seconds if anyone was still adding the poll or sort of adding information or feedback. And I hope to see everyone who joined the call today in the next session. So we'll let you know what sort of topic we'll be focusing on. So thank you so much.